I'm Simon Ndirangu. I'm from Kenya Medical Research Institute in Kilifi in Kenya, and I'm going to talk about measurement of drug levels in clinical trials. So in some clinical trials, they include a pharmacological component. That is where a drug is given, and then we need to measure drug levels. And the measurement of drug levels is always important, and this is either done in biological matrices. It can be whole blood, serum, or plasma. And when you are taking samples for such measurements, it is important to ensure that, first of all, that the correct dose of the drug is given, the samples are taken at the right time, and samples are collected in appropriate tubes. And also, it is important to make sure that after collections, immediately after collection, they are stored at the right temperature, either in the fridge or in the freezer. You also have to be sure that the method which you require for measuring these drug levels is also available before you start the study. So you need to talk to the people who can help you in that. I want to talk about a bit of two concepts which are important when you do clinical trials involving drugs. The first one is pharmacokinetics, and the other one is pharmacodynamics. Pharmacokinetics involves the study of what the body does to the drug. Once you take a drug, what happens to the drug in the body? And there are three processes which are important. The first one is absorption, and all you need to know here is that the question we are addressing is the drug getting into the body after you take it? The second one is distribution. Once the drug is in the body, it goes to other parts of the body after you've taken it. So we have to know where it is going. The third mechanism is metabolism and elimination. And once you take a drug, the body treats the drug as a poison, so it finds a way of getting the drug out of the body. So those three mechanisms are important to consider when you take a blood sample. The other concept is pharmacodynamics. I'm not going into details in this. All it means is that once the drug is in the body, what is it doing? You take a drug to have some effect in the body, let's say to treat some disease and so on. So that effect can be either efficacy, that means the drug is effective in treating your disease. You can also have toxicity, so that's also associated with the drug. So a drug can be either good or bad, and most drugs have both effects. And also you can have other side effects which are associated with the drug, such as you might have rashes, itching, and so on. So why is it important to measure drug levels? The first thing is that in some clinical trials, they involve drug development. So early in the clinical trial, you actually have to find out whether you are getting the right dose and whether the dose that is suggested for use in humans is going to result in adequate concentrations in the body. So we need to measure that. In other cases, other clinical trials, the drug is already being used in humans, but you want to know whether, if you are using it in children, whether the dose which is recommended is appropriate for that special group. Other special groups include pregnant women. You may also have patients who have malnourishment in some resource poor settings. So, and you may have others who have several other comorbidities, other infections, other than just the disease that you are interested in. So all these factors interact to give some effects of the drug. So you really have to know whether there is any change in the concentration. And once you measure the drug concentration in the body, it is going to help you to optimize the dose, to ensure that the dose you are giving is appropriate for that population or for that person. And you also establish the therapeutic range, the concentration of the drug which is effective and which is not too much or too low for that patient. So those two factors are going to be important. The other thing is that in some cases there are serious consequences if there's overdosing, especially if you are giving a drug through the intravenous route, or in some other cases, you might underdose the patient, so the patient doesn't get any benefit because the drug was too lethal. Some drugs have what is known as a narrow therapeutic or toxic index. That means the difference between the concentration which is effective and that is which is toxic is very narrow. So for these drugs, you need to measure the levels in the body to ensure that you can monitor whether that patient is having the right concentrations. In some other cases, there is altered physiology, especially in pregnant women or patients who are very sick. So in these cases, the way they handle the drug in the body may not be the same as that in healthy adults or other populations. So in this group, you also need to do measurements to ensure that the dose you are giving is adequate, is not having serious side effects. And in other cases, in most cases, when a patient is sick, they are treated with very many drugs at the same time. You may be having severe malnutrition, you are malnourished. At the same time, you have bacterial infections, so you need to be given drugs for treating the bacterial infections. And you may also have high fever, you are given paracetamol or aspirin to reduce the fever. So there might be drug-drug interactions. And in all these are going to affect how the body handles your drug. The other thing is that some patients, especially those who are given drugs to go and take at home, may not take the complete dose. They may miss some of the doses and skip them. And it is important to measure the drug levels and see whether they have adequate concentrations. Because in some cases, you find that those people who are on long-term medication like in epilepsy or HIV/AIDS, they have to take drugs every single day for the rest of their life. So at times they get fatigued by the drugs and sometimes they don't take all the drugs. And that can lead to inadequate concentrations. So once you measure those the levels in these patients, you can be able to know whether they are taking their drugs appropriately. Some of the drugs which are usually monitored are anticonvulsants or antiepileptic drugs. These are drugs which are used for treating seizures or convulsions. Others are antibiotics such as gentamicin and ciprofloxacin. We also have anti-cancer drugs. Anti-cancer drugs are very toxic, so it is important to know that the dose that is used in a certain patient is not too toxic. And recently, there's a lot of work which is going on antiretroviral drugs. These are drugs which are given to treat HIV/AIDS, and they are usually given not just one drug, several of them. In some patients they might be taking 3 or 4 more drugs and very many pills at the time. So those are some of the patients who may not comply with the treatment regimen, and we need to check to ensure that they are taking the drugs in the right way and the concentrations they get in the body are adequate. Otherwise, we are going to end up with multi-drug resistance, which is very serious, especially for HIV/AIDS. Why should we measure drug levels? In some cases, we have said, as I've just said, if a drug has a narrow therapeutic index or range. In some other cases, if you give a certain dose of a drug, there's no good relationship between the drug, the dose of drug given, and the plasma concentration which is obtained. In some other cases, there's a good relationship between the plasma concentration and the therapeutic or toxic effects. So you need to establish which one of these categories you are dealing with. In some other cases, the drug shows what is known as non-linear pharmacokinetics. That means if you double the dose, it doesn't mean the concentration in the body is going to increase by 2. And this is because there's what is known as saturation of the enzymes which handle the drug. And a good case is phenytoin, which is an anticonvulsant. In some other cases, it is very difficult to interpret the signs and symptoms of toxicity once the patient has taken the drug or therapeutic failure and evaluating therapeutic responses. So you need to do to measure drug levels in this group of patients. And what methods do we use for measuring drug levels? There are several methods, but some of them are not very specific. These include ELISA. These are bioassays. We also have radio immunoassay and fluorescence polarization immunoassays. Some of these are very, they have advantages in that you can do them very rapidly, like FPIA. But the most popular methods which are commonly used include chromatography, high performance liquid chromatography, because you can be able to do a lot of drugs. Most of the drugs are stable at room temperature and in the matrices, and you can be able to use chromatography. And more recently, mass spectrometry is now the gold standard. In developed countries, it is available. It is very high sensitive and selective. The only disadvantage is that it requires highly trained personnel and is very expensive to buy and also to maintain in these poor countries. So that is not one of the options that we use. So when you want to measure drug levels, you need to consider the cost that it is going to incur, because everything costs money. And there are several categories of costs which you need to consider here. The first is economic consideration. You need to have a laboratory to be able to do your analysis. Once you have the laboratory, you need the equipment, the lab equipment. You need to buy the equipment, maintain the equipment, and also that equipment is going to age with the time. So you have to factor in all those costs. The other thing you need to do is you need to buy laboratory reagents. And these are high purity solvents and also analytical standards. You also need to train people to help you. And after that, you may consider charging a user fee to recover the cost, because it's going to be expensive. So people whom you are doing the assays for, they have to pay for that. And the other factors you need to consider once you are doing a clinical trial: how many drugs are you going to monitor? Is it the parent drug and the metabolite, or just metabolite? And how many patients per sample do you need to take? And how much blood? Because of ethical issues, you need to make sure that the sample is kept to the minimum. And where are you going to do the analysis? In these poor countries, you may not have the lab. You might have to go to another collaborating institution in a developed country. So you have to consider that. And other factors which you need to consider when measuring drug levels, these are those which are related to administration of the drug, pharmacokinetic principles, whether the drug is absorbed properly, distribution and metabolism, and other analysis factors. Factors that may affect the concentration of drug that is measured and how it is interpreted. This include disease states, malnutrition, renal failure, hepatic problems, and also cardiac problems. Also habits, people's habits like diet, smoking, and alcohol, those may interfere with the drug metabolism. Women who are pregnant, they may not handle the drug like others because there are physiological changes. And other patients don't take the drugs. Then you may have drug-drug interactions. Then some drugs are also highly bound by protein, and others are poorly bioavailable. That means if we give them orally, very little of the drug goes into the body, and how the samples are taken for measuring the concentration. Factors that are important, they include drug administration and blood sampling. Sometimes the wrong dose may be given, or the dose is skipped. The patient doesn't take the dose because the patient was undergoing a procedure, or the nurse forgot to give the drug because maybe they were doing something else. Then the dose is given at the wrong time. Some drugs you need to give them, let's say, twice a day or 3 times a day. In some cases, it might be just given once. And in other cases, the dose is given at the right time, but the sample is not taken at the correct time. Then drugs which are given through infusion, sometimes the rate is given is too fast or too slow. And in infusion, sometimes is stopped because another procedure needs to be done, or the nurse is giving another drug through the same line that they should have taken the drug. And sometimes the sample is taken from the same vein where the drug is also given. And this is especially for drugs which are given intravenously. So in this case, you need to fix two IV lines, one for drug administration, the other one for taking the sample. Other factors that may affect pharmacokinetics, these include appropriate weight, especially people who are obese or who have lost some weight. Then sometimes there might be changes in the liver and the kidney. Other changes might occur because the percent of drug which is bound changes because you have the plasma proteins, they have changed. And then the enzymes that are handling the metabolism may change. The other factor which you need to consider is to do with the laboratory where the drug assays are being done. Sometimes the assay is not the correct one. Sometimes once the assay is done, while transferring data or entering data from the lab book, then mistakes are made. Or the data is entered in the wrong way. Let's say you had samples where a sample was taken at 1 hour, another one at 4 hours. You need to enter the data in the right order according to how samples were taken. Then sometimes it is the active metabolite which is required but was not measured. Then there are interferences with the assay by other drugs or endogenous substances, or the wrong specimen was taken. That means you should have taken serum, but you end up bringing a blood sample to the lab or urine sample. Those are the wrong ones, so you have to be clear. Then what happens is that in some cases, even when the drug is already being used in the market, you might want to do what is known as therapeutic drug monitoring. And the reason here is important because sometimes you want to be sure that the drug concentrations, they correlate with clinical response. And this is important especially for antiretrovirals. In some cases, there's a lot of variability in the, between patients or individuals in how they handle the drug. Then you can use the therapeutic drug monitoring for managing drug-drug interactions. And also, therapeutic drug monitoring, they show that the patient is not adhering to treatment. And finally, therapeutic drug monitoring is a tool for managing concentration-related toxicities. So you take a sample, you measure. If the levels are too high, you can reduce the dose in time. But there are many challenges that are faced when you are measuring drug levels, especially in clinical trials. Sometimes the people who are setting up the trial do not understand the principles of pharmacokinetics. So they take the wrong samples, they take them at the wrong time. Then at other times, once the sample is taken, it is stored at the wrong temperature. So the drug ends up degrading. Sometimes there is no history on what else the patient was taking, which other drugs the patient was taking, so that we can be able to know whether there was any interactions or possible interactions. And then failure by the investigator to consult an expert, usually a pharmacologist, when deciding the study. So we may end up having the samples, but all of them are not going to be useful.